Okay, Assalamu alaikum. My name is Immad Ashraf. I'm a second year medical student, and today we'll be having a pulse session on two of the lectures in the GIT block, gastric secretion and gastric motility. So, first of all, I want to ask you guys, anyone in the meeting right now, what do you think of GIT so far? Do you like it? Like, is it tough? Anyone can answer? Guess no one wants to answer. Okay, so gastric secretion, there's a lot of things that you need to know in this lecture. And fortunately, you do not need to memorize too much as long as you use your logic and as long as you know a few core concepts. And inshallah, I'm going to try and make it as easy as possible so that you don't have to spend too much time on this lecture. So I think we should get started so we can finish early. Okay, so the first thing that we have is the gastric juice, right? It consists of four things: hydrochloric acid, HCl, and this is used for protein digestion. You have pepsinogen, which is also used for protein digestion. You have an intrinsic factor, which helps in the absorption of vitamin B12 in the ileum. And you have mucus, which protects the mucosa of the stomach from the HCl and lubricates it, prevents ulcers. So hydrochloric acid actually breaks down the proteins, and then pepsinogen breaks the peptide bonds. And we'll study more about this in the next few slides. But these are the four main components of gastric juice. And they're quite easy, so there isn't much to go over in this slide. Now, we'll talk about the cell types, and this is just a quick summary. We're going to come back to these cells throughout the lecture. So if you can remember, try to remember all of these right now. That would be great because it will help you rearrange your thoughts during the lecture. So the first one is the parietal cell. It's located in the body of the stomach, and it produces hydrochloric acid and the intrinsic factor. Then we have the chief cells, and these are also located in the body, and they produce pepsinogen. Pepsinogen is turned into pepsin. Pepsinogen is its inactive form, and we'll study about this later, but you can just keep that in your mind. Then we have G cells, which are in the antrum this time, not in the body, and they produce gastrin. We have mucosa cells that are also in the antrum. They produce mucus, you can tell by the name, and pepsinogen. However, the main pepsinogen producing cells are chief cells. The antrum is distal to the body, and you probably have taken this in anatomy, or you will take this in anatomy, but the antrum is distal to the body of the stomach. So these four types of cells are very important, and they form the backbone for this lecture. So you can look over it, and it'll help you if you just remember these right now, because I'll refer to these throughout the lecture. So I'm going to give you like 30 seconds to look over it. Okay, I guess that's enough. Now we move on to the next one. Okay, we have two types of glands in the stomach. They're oxyntic glands, which are these over here, oxyntic, and pyloric. And the only thing that differs in these glands are their location. So the oxyntic glands are in the body. So this is where oxyntic glands are. And the pyloric glands are in the antrum, which is over here. As you can see, it's slightly distal. And so these glands, they share some types of cells, and some types of cells are different, but the main difference in these glands is their location in the stomach. So these are the two main types of glands. Okay, now we will talk more about the oxyntic and pyloric glands. So most of the pictures I've taken are from Dr. Abdul Jabbar's lectures, which he took from Dr. Peter's lectures. And so I'm going to use them because at the end of the day, he's giving the questions, and it would be better if I could explain how he would want to. So Okay, we have oxyntic and pyloric glands. Pyloric are on the right and oxyntics on the left. So the stomach is lined by simple columnar epithelium throughout. It doesn't matter which part of the stomach it is. It's lined by simple columnar epithelium. We have mucus cells, and an example of a mucus cell is this cell right here, which I'm going to circle. That's a mucus cell. And these obviously produce mucosa. And the function of mucosa is to protect the gastric cells, which like to protect themselves, the gastric cells, from the hydrochloric acid and from other corrosive substances in the stomach. It's to prevent ulcers, bleeding, and if anything goes wrong in this, you can have pathologies like ulcers and other things that we'll study about later. Then the mucus cells dip inwards to form gastric pits. So this whole thing is a gastric pit. And you can see it's dipping inwards like a U. And all of these cells are mucus cells, obviously. Not all of them, but the ones in this brown color, which I've circled, are mucus cells. So when they dip inwards, it's called a gastric pit. And these gastric pits have the different types of cells. So we have parietal and chief cells, which we talked about earlier in the table. They have a turnover of 90 to 120 days. And the surface mucus cells have a turnover of 3 to 5 days, which means these cells right here are replaced every 3 to 5 days, but the other cells, the specialized cells, are replaced every 3 to 4 months, which is 90 to 120 days. We also have a progenitor zone, which is indicated over here and over here. So you see these two types of glands are different. These are oxyntic glands and these are pyloric glands. And as we learn over here, oxyntic is in the body and pyloric are in the antrum. So the oxyntic glands have their progenitor zone superior. You can see the gastric pit is just starting. It's barely dipping, and you already have a progenitor zone right here, and the stem cell is within the progenitor zone. So the stem cells can develop into any type of specialized stomach cell depending on what's required. The pyloric glands, on the other hand, have the stem cells more distal. So this is the stem cell in the progenitor zone of the pyloric gland. As you can see, it's much deeper. There's this difference between them. And that's a characteristic difference in the oxyntic and pyloric glands. I noticed Dr. Abdul Jabbar explained all of these concepts on one slide. However, I tried to break it down into different slides because I feel like it's less hectic that way. So let's move to the next slide. This is literally the most important slide in like both of these lectures, because this forms the basis for every other topic in this lecture. So we have the gastrin-histamine axis. And the gastrin-histamine axis is the major pathway for production of acids by the parietal cells. So parietal cells produce HCl. Gastrin is produced by G cells, and gastrin basically stimulates the production. And we'll see how. We also have EC. So this is the parietal cell, the blue one. And we have an ECL, which is an enterochromaffin-like cell, which is this small green cell right here. This produces histamine. So let's talk about the whole pathway, the gastrin-histamine axis. So the G cell releases gastrin. Now, why does it release gastrin? There's many reasons. One of them is if we have GRP, gastrin-releasing peptide, and we'll talk more about the reasons later, but I'm just giving you a quick overview right now. So the GRP could lead to the release of gastrin. Ingestion of food could release gastrin. And there's a bunch of other factors that could release gastrin. Now, when gastrin is released, it's secreted into the blood. So it's a hormone that's secreted into the blood. And when it's secreted, it activates the enterochromaffin-like cells. So the enterochromaffin-like cells, when they're activated, it leads to release of histamine because that's what they produce. And then histamine uses paracrine signaling to activate the parietal cells. And when the parietal cells are activated, they release HCl. So as you can see, there's a lot of cells involved. So let's write them on the side over here. So the first one are G cells. Okay, they produce gastrin, which goes in the blood. Gastrin then acts on ECL, enterochromaffin-like cells, which release histamine. Histamine then has a paracrine effect on parietal cells, and parietal cells release HCl, hydrochloric acid. And hydrochloric acid, as you know, breaks down the protein into smaller peptides. and basically it produces an acidic environment that's required in the stomach. In case you do not know what paracrine signaling means, it's a type of signaling in which nearby cells are stimulated to produce products. So it only works on nearby cells. And as you can see, the ECL and the parietal cells are present in the gastric pit. So they're present close to each other. So so in this case, paracrine signaling is more than enough for the communication. We also have chief cells, which are these purple cells over here. And they release pepsinogen and lipase. Pepsinogen is the inactive form of pepsin, and we will learn later how it's activated into pepsin. And lipase, obviously, is something that breaks down fats, right? If anyone has a doubt, you can stop me at any time. You can unmute if you want, because there aren't too many people right now. So, and then I can explain anything that you want me to. So don't be afraid to stop me at any moment, please. But that's it for this slide. And again, this is the most important slide, gastrin-histamine axis, because a lot of the things that we learn after this are based on this concept. So let's go over the order one last time. G cells, gastrin. Gastrin goes to the blood. From the blood, it activates ECL. ECL releases histamine. Histamine acts on parietal cells, and parietal cells release HCl. And that's the gastrin-histamine axis. Now we have D cells. And these cells are these yellow cells over here. And so the D cells are called delta cells, and they produce something called somatostatin. And one important thing that you need to remember is somatostatin is an inhibitor. So anytime you hear of the word somatostatin, you want to immediately think it's an inhibitor. It's inhibiting something. And it is not only an inhibitor in the GIT, it's a general inhibitor for endocrine glands, all of the endocrine glands, and there are so many. You'll learn them in EndoRepo. But just you need to know that somatostatin is an inhibitor for a lot of them, including the glands that are present in the stomach. So they inhibit paracrine secretions and gastric acid secretions. So this is an inhibitor, and the other ones were the gastrin-histamine axis that was stimulating acid secretion. Delta cells have axonal extensions, which means they have neurons that allow them to communicate with other cells and inhibit their secretion. So they have neurons that are part of the autonomic nervous system, and that allows them to communicate with other cells. You don't need to know whether they're autonomic or any other type of neuron. Just know that they have neurons that allow them to communicate with other cells. So, for example, the D cells, or the delta cells in the pyloric glands, they have extensions on G cells, so they can inhibit gastrin. Remember, somatostatin always inhibits. Cell extensions, which the delta cells have in accentic glands, are on the parietal cells, ECL cells, and chief cells. So parietal cells means they can inhibit HCl. ECL means they can inhibit histamine, and chief cells means they can inhibit Chief cells means they can inhibit, wait, give me one sec. Chief cells means they can inhibit pepsinogen, right? By the way, as I said, that table is super important because all of these cells are going to be mentioned again and again. So chief cells, pepsinogen. So it can inhibit all of this because it's an inhibitor. Oops. Okay, now we have enterochromaffin cells. So remember how we said we have, remember how we said we have ECL, enterochromaffin-like cells, but these are enterochromaffin cells, so they're not the same. Please do not confuse them because they have the same name, but the like is very important because ECL releases, as you see, somatostatin. The EC, or enterochromaffin cells, release serotonin. So serotonin is also called 5-hydroxytryptamine. That's why you see the 5-HT. And they can give you either term in the exam. And so you should remember that serotonin also means 5-hydroxytryptamine. So it modulates gastric acid release in the stomach. It also has a very important modulating role. And we'll study about that in the next few slides. So any doubts so far? Anything you want to ask me so far? Because we're done with this section of the lecture. So if anyone has any doubts, you can ask me now before we move on to the next part. Okay, so now we have the major constituents and function of the gastric juice. So we already talked about the constituents of the gastric juice, the first four, earlier. So now we'll go into a little bit of detail about them. The intrinsic factor is released by parietal cells, and it binds vitamin B12. It's necessary for the absorption of vitamin B12 in the ileum.
Immad?
Yes?
Sorry, there's someone in the chat. They're asking to re-explain the serotonin part.
Sure. So enterochromaffin-like cells, ECL, enterochromaffin-like cells, produce somatostatin. And somatostatin is an inhibitor for, in general, everything. It inhibits everything, anything that you can think of. Enterochromaffin cells, not like just enterochromaffin cells, produce serotonin, which is also called 5-hydroxytryptamine, and that's why you see the 5-HT in brackets. And this 5-hydroxytryptamine serotonin, it also modulates gastric acid release in the stomach. It doesn't always inhibit it, but it also regulates it. And there's a whole pathway about this. It's called the calcitonin gene-related peptide pathway, and we'll study about that in the next few slides. So for now, just remember that ECL and EC are not the same cell. They're different cells that produce different things and have different functions. The pink one, which you see here, is an enterochromaffin cell. The green one over here is an enterochromaffin-like cell. I hope that makes it clear. Okay, as I was saying, let me repeat now. The intrinsic factor is released by parietal cells. It binds to vitamin B12, and it's necessary for its absorption in the ileum. This thing, surprisingly, is the most indispensable part of the gastric juice. Everything else we can make do without because we have the pancreas, and the pancreas produces most of these things as well. But the intrinsic factor is only produced in the stomach, and it's indispensable. Without it, it would be impossible to live because vitamin B12 is very necessary for us. Then we have the hydrochloric acid, which is also released by parietal cells. So the parietal cells releases intrinsic factor and hydrochloric acid. And so as I said earlier, pepsinogen is the inactive form of pepsin. So pepsinogen is converted into pepsin by HCl, which is acidic. So when HCl acts on pepsinogen, it cleaves the part that is required for catalytic conversion into pepsin. And once the HCl cleaves that part, then the rest of the pepsin can automatically, the rest of the pepsinogen can automatically convert into pepsin via HCl. So HCl is very important for the conversion of pepsinogen into pepsin. Hydrochloric acid also denatures ingested proteins, which makes it easier for the pepsin to break the peptide bonds, and it's also bacteriostatic or bactericidal. So any of the bacteria that we ingest along with our food, the hydrochloric acid can kill it because its pH is too low for bacteria to survive. Then we have the pepsin released by the chief cells. Well, technically, pepsin is not released by chief cells. Chief cells release pepsinogen, and then pepsinogen is converted into pepsin by HCl, but just for the sake of simplicity. And we also have protein. The pepsin also hydrolyzes protein. It's not as essential as pancreatic proteins, which break down. It's not as essential as pancreatic proteases because those are generally stronger than pepsin. So even if we have a problem where there's no pepsin in the stomach, it really does not affect the quality of life that much, just because we also have proteases that are present in the pancreas that help breaking down proteins. It is also a bactericidal. By the way, bacteriostatic is something that stops bacterial growth and then lets the immune system deal with it, and bactericidal is something that just kills bacteria. You don't need to know that, but I just thought I'd say that. Gastric lipase is released by chief cells, and it hydrolyzes triglycerides into diglycerides and fatty acids. So it basically breaks down fats. It's also not essential because we have something called pancreatic lipases, and they can break down the fats just as well as gastric lipase, if not better. So we don't really need gastric lipase. Then we have the mucus, which is released by mucus cells. It also releases bicarbonate. So bicarbonate, which is chemically HCO3 minus, is also released by the mucus cells, and it acts as a chemical barrier against the luminal acid. So it neutralizes the acid so that the acid does not affect the mucus cells themselves. And it also acts as a physical barrier because it provides lubrication. So that's it. And as I said, you see all of these, most of the constituents are also produced by the pancreas, which means they're not essential for life, but the intrinsic factor is only produced by the stomach, which makes it indispensable. Like we need it to live. If that's clear, I'm going to move on to the next part. Okay, so now we're getting into like the real part of the lecture, quote unquote, because this is where all of the ion channels and ions start to come in. But don't be scared because it's actually very simple once you understand it. You actually don't need to remember it if you just understand a few concepts. So this is a parietal cell. A parietal cell produces HCl and the intrinsic factor, but we're not worried about the intrinsic factor right now. We're just going to focus on HCl. So this is a resting parietal cell, which means it's not producing HCl, and this is a secreting parietal cell, which is producing HCl. So the resting cell has a cytoplasm that's occupied by tubular vesicles, which contain ATPases. ATPases are obviously something that works on ATP, and tubular vesicles are basically just storage. Okay, just remember that for now. The high energy demand of secretion can be overcome due to numerous mitochondria. So the parietal cells obviously have to secrete so much stuff, and they require a lot of energy for this because ATPases require ATP. So how is this demand fulfilled? It's due to the mitochondria. They have a lot of mitochondria, and it's actually 1/3 of their cytoplasm is mitochondria. So they have a lot of mitochondria. We also see canaliculi, which are these things right here. I'm going to draw around them so you can focus on them. Those are canaliculi, and canaliculi are deep enfoldings which increase the surface area of a cell, and they're usually present at the apical surface. So they increase surface area. That's what they do, and you'll see why this is essential in a bit. So now we're going to move on to the secreting cell, which is a cell that's actively producing HCl. The first thing you can notice from the picture itself without even reading anything is that look at how much it has changed. Like the one on the right, this one, looks nothing like this. Like they look completely different. So the parietal cell can produce acid against the gradient, which means, and this is very important because obviously we already have HCl in the stomach, right? But we still want more. We want more. So the parietal cell, against the gradient, it can produce HCl. So it obviously undergoes morphological changes because it looks completely different. The tubular vesicles fuse with the canaliculi, leading to an increase in surface area and increase in microvilli length. So I want you to focus on this now. Look at this. That is 1 microvilli in the resting state. And look at this. That's a microvilli in the secreting state. You can see how much longer it's gotten. And you can also see how much the surface area has increased. Earlier, this was the surface area, this much, basically only the top part. But now we have all of this. All of that is surface area now. Let me just clean this up a little because it looks very messy. Okay, so yeah, it increases surface area and it increases microvilli length. And both of these are to increase surface area. Now, due to these changes, the parietal cell is able to release large amounts of HCl into the gastric pit. So because of the increase in surface area, there's more area to work with, and this increases HCl production. Now, how exactly is HCl produced? So this is the regulation of HCl production. So how does the body know you need HCl in the stomach now? It's quite simple. Well, it's not simple, but it's not hard either. So we're going to talk about the 3 main factors that lead to an increase in HCl, which stimulate HCl. So stimulation first. The first one is histamine, which is over here. Histamine, okay? Then we have acetylcholine, and lastly, we have gastrin. So all of these act on different receptors. Histamine acts on the H2 receptor. As you can see in the diagram, this is the H2 receptor. Acetylcholine acts on the M3 receptor, which is this receptor over here. That's the M3. M3 is a muscarinic receptor. And gastrin acts on the CCK2 receptor. So you need to remember these, and you need to remember both the hormones or whatever, or these ligands, the hormones or ligands or neurotransmitters. So histamine, ACH, gastrin, you need to remember all three, and then you also need to remember the receptor they act on. Another important thing to remember is if all of them were secreted together, their effect is more than it would be if it was additive, which means if histamine was to act on H2 and have an effect, suppose, like, let's just say the effect was 2, and if ACH acts on M3, let's say its effect is 6, and if gastrin acts on CCK2, its effect is 4. So if all of them act together, it would be more than 2 plus 6 plus 4. So like if this is equal to 12, if they all act together, they would have an effect that's equal to 20, for example. So if they all act together, the effect is going to be much greater than if they act individually. Another important thing, and this is very important, by the way, is what they increase inside the cell. So now that they've acted on the receptor, what intracellular changes come about because of this interaction between the ligand or the hormone or the neurotransmitter and the receptor. So histamine increases the cAMP level, which is cyclic AMP, whereas ACH and gastrin increase calcium levels. And you can see this in the diagram as well. Honestly, this entire lecture can be explained on like diagrams only, and that's what I love about GIT. So histamine increases cyclic AMP, and muscarinic and ACH and gastrin increase calcium. So when increased calcium and cAMP are present intracellularly, it leads to the fusion of tubular vesicles with the canaliculi. And obviously, we studied in the earlier slide that leads to an increase in surface area. In addition to an increase in surface area, it also activates potassium channels. So when cyclic AMP and calcium fuse, they activate potassium channels. The potassium channels are required as they are used for potassium hydrogen ATPase. So this guy right here, okay, the potassium channel is used because it helps in the activation of the potassium hydrogen ATPase. And why is this important? It's because hydrogen then can then leave into the lumen. This hydrogen can exit the cell and go into the lumen because of this hydrogen potassium ATPase. This is an antiporter, and an antiporter is basically something that exchanges two ions. So potassium is going into the cell and hydrogen is going out, and the charges are balanced, and that's why it works, because they're both 1+. So the hydrogen leaves the cell and the potassium comes into the cell. Now we have the hydrogen in the cell. We also have chlorine channels, which are not shown on this image, but they're shown in the next. Okay, actually they are shown right here. These chlorine channels are called CFTRs, which is, the full form is, I'm going to write it on the top right, cystic fibrosis transmembrane regulators. So CFTRs allow the chlorine, to leave the cell and move into the lumen. Now we have this H+ and this chlorine in the lumen. So what are they going to do? They're going to combine and form HCl, which is what we wanted because our parietal cells forms HCl, right? So through these three hormones / neurotransmitters / ligands, we eventually have HCl formed. And we have the potassium channel, that's very important, this, the first one, and then we have the chlorine channel, that's very important. The hydrogen potassium ATPase, the proton pump, is also very important. So this was stimulation. This is how hydrochloric acid is formed, but how is it inhibited? What if we don't want hydrochloric acid in the body? We want less hydrochloric acid. Then we have somatostatin. I told you to remember that somatostatin always inhibits. It's an inhibitor. Its only function is inhibiting. So somatostatin inhibits the formation of hydrochloric acid, and so does prostaglandin E. You don't need to know where it comes from, the prostaglandin E, just remember that it inhibits. Somatostatin inhibits intracellular CEMP levels. So this cyclic AMP that was increased due to the histamine, the somatostatin is going to oppose it. It's going to decrease the cyclic AMP, and it also inhibits G cells. Do you remember what G cells form? They form gastrin, which stimulates the production of hydrochloric acid. So somatostatin inhibits the intracellular cyclic AMP and G cells. So that's how it inhibits acid production. And prostaglandin E also inhibits acid production. So if you have any doubts, you can tell me now before I move on to the next slide, because the next slide is more ion channels, but these are the last ones in the lecture, don't worry. So if anyone has any doubts, you can put them in the chat now. By the way, I can't see the chat, so if anyone does have a question, I'm sure someone will read it out for me. Okay, I guess no one has any doubts right now. So we'll move on to the next one. I hope this is clear because it's also very important. Okay, now we have the ion channels. So we have a lot of ion channels, but if you treat it like a story and not as just like remembering it, then it's way easier to remember. So we're going to go step by step. The first one is the sodium-potassium ATPase. And what does it do? It's an antiporter, which means it exchanges. So sodium goes out of the cell and potassium comes into the cell. Okay, as you can see, it's very clearly shown. Then we have an anion exchanger. And an anion exchanger obviously exchanges two anions. So chlorine is going into the cell, so chlorine is in, and bicarbonate or HCO3 minus is out. So now we have potassium and chlorine inside the cell. Do you remember this? This was in the last slide over here. This one. So the H plus slash K plus ATPase is used to recycle potassium back into the cell and secrete hydrogen into the lumen. So we have this potassium inside the cell, and we have this hydrogen inside the cell. And I'll tell you how the hydrogen gets into the cell, but know that we have both now, and we have this thing. This is a potassium channel. It's not an ATPase, it's not a pump. The H plus, also one more thing, the H plus slash K plus ATPase is also called a proton pump. So this channel, this one is not a proton pump. It's a simple channel in the cell membrane that allows potassium from inside the cell to leave and go into the lumen. And the only reason it does this is so that this potassium can then be exchanged for hydrogen. This potassium, when it leaves through this potassium is then going to be shuttled into the cell using the proton pump, and the hydrogen is going to be shuttled in the opposite direction into the lumen. So now we have a hydrogen in the lumen. Can we agree on this? We have one hydrogen in the lumen. It's waiting. So then we have now, do you remember the anion exchanger brought chlorine into the cell, right? So now we have chlorine in the cell. We want this chlorine to leave the cell, and that's where we have CFTR, which is cystic fibrosis transmembrane regulator. This is also a channel, and it's not a pump, it's just a channel. It's an opening in the cell membrane that allows chlorine to leave. So this chlorine leaves the cell through the CFTR, and now we have one chlorine, right? So now we have one hydrogen and one chlorine outside in the lumen. What are they going to do? They're going to react, and they're going to form HCl. And that's the hydrochloride that's produced by parietal cells. And that's how hydrochloride is produced. But there's a few other things that we need to talk about. So you notice how this bicarbonate is leaving the cell, is going into the blood, because chlorine is coming into the cell. So eventually, in the gastric system, there's going to be so much hydrocarbonate in the blood that the blood is going to become alkaline, because HCO3 minus is an alkaline substance. It's not acidic, it's alkaline. And this is called alkaline tide. So an important question is, what is alkaline tide? Alkaline tide is the blood, is the venous blood actually, in the gastric system. And the reason for it being alkaline is because of all the bicarbonate that's entering the blood due to the anion exchange. The anion exchange is taking chlorine from the blood and giving it HCO3 minus. And keep in mind that this is venous blood, not arterial, venous. We also have this transporter over here, NKCC1. It doesn't have a full form, and it's called NKCC because it transports 1 sodium, 1 potassium, and 2 chlorine together. So it's not an antiporter like the other two. Antiporter means opposite directions, like this. This is called a co-transporter or a symporter. And in this case, both of them move in the same direction. And the charges are balanced because sodium and potassium means 2 positive and 2 chlorine means 2 negative, which is 0. So this NKCC1 acts as an excess source for chlorine. So you can see the chlorine in the diagram. It joins the chlorine coming from the anion exchanger and exits through the cystic fibrosis transmembrane regulator, which is a chlorine channel. Okay, remember how I said we have hydrogen in the cell, and we'll talk about how it got there later. So this is how it gets there. So we have CO2 in the cell, and CO2 is a metabolic product because a cell is always going to produce CO2. That's due to the metabolism, due to the mitochondria, all of that, it produces CO2. And we also have water in the cell, H2O. Every cell has water. We need water to survive. So both of these join, these get added, and they form H2CO3. And the enzyme that's responsible for this reaction that catalyzes it is carbonic anhydrase, which is Ca, as shown in the diagram. So Ca catalyzes the reaction of CO2 and H2O to form H2CO3. But H2CO3 is not stable, and that's why it breaks down into 2 parts. This is the first part, HCO3 minus, and the second part is H plus. So HCO3 minus is going to be exchanged with the anion exchanger, and H plus is going to be exchanged with the H plus K plus ATPase, also known as a proton pump. And that's how the H plus comes into the equation. Earlier, I told you to forget about it, but now we see how we got this H plus in the cell, right? So now I explained this earlier, but an important question asked is, we already have 1 potassium. Let me clear this up a bit so that it's more clear for you guys. Okay. So one important question you may ask is, we already have this for potassium, right? Why do we need this? Why do we need 2? And the reason we need this channel over here is because this allows the potassium to leave the cell, and then the same potassium that left the cell is exchanged and comes into the cell in exchange for some hydrogen. This hydrogen would not be able to leave the cell into the lumen if it wasn't for this potassium already outside. Because remember, it's a co-transporter. That means one needs to go this way and the other needs to go this way. Well, the one going this way is the potassium. And for it to go in this direction, it needs to be outside. And this channel, this one, allows it to be outside. So that's it. I promise you, this is the hardest. This, on first glance, looks like the hardest slide in the lecture, but it's not that difficult, as long as you know where everything is going. So that's why I'll go over it one more time really quickly, because I believe that repetition is the best thing for topics like this. Really quickly, just one more time. So we have this sodium potassium ATPase. Potassium comes into the cell, sodium goes out. We have an anion exchanger. Hydrogen carbonate is going outside and chlorine is coming inside. Because hydrogen carbonate is going outside, we have alkaline tide in the venous blood, because HCO3 minus is alkaline. The chlorine exits through the CFTR, which is a chlorine channel, and enters the lumen. The hydrogen exits via the H plus K plus ATPase. We have a K plus outside because of this channel over here that allows K plus to exit. Where do we get this K plus from? It's from over here. It was exchanged for sodium. This potassium then enters the cell and the hydrogen goes the other way, which means we have one H plus and one Cl minus, which means we have HCl outside. Now, we need to talk about how the H plus came about. So we have CO2, which is a metabolic byproduct, and we have H2O, which every cell has. Carbonic anhydrase is going to form H2CO3, and this is unstable. So it's going to split into HCO3 minus, which is used by the anion exchanger. And we have H plus, which is used by the proton pump, or the H plus K plus ATPase. Then we also have the NKCC1, which is a co-transporter. It allows one sodium, one potassium, and two chlorine to come into the cell, all of them in the same direction. And this chlorine then joins the other chlorine that came into the anion exchanger and exits to the chlorine channel. That's everything that we need to know on this slide. Are there any doubts? Because this is an important slide and it will be tested in the exam. If you have any doubts, please put it in the chat and I'm sure someone will read it out to me. No doubt, I guess. That's great. We'll move on to the next one, and it gets much easier from here, I promise. We have gastric pepsin. Okay. Pepsin is an endopeptidase. Endopeptidase. And endopeptidase is basically something that breaks peptide bonds, but only when they're between two peptides. So if this is, let's say this is 3 peptides, and we have this bond, we have this bond, and we have a bond over here. Endopeptidase is only going to break this bond. It's not going to break this bond. And because it's an endopeptidase, when it breaks this bond, it's going to leave this peptide and it's going to leave these 2 peptides. So basically, an endopeptidase leads to a mixture of peptides, which are of different lengths. Pepsin is stored in the form of pepsinogen, and it's activated at pH 4. pH 4 is an acidic pH, and the reason for the pH being 4, obviously, is HCl. And once we have the pH 4, it causes an autolytic cleavage, hence exposing the catalytic site. So this pH, which is low, it cleaves a part of pepsinogen. So let's say this is pepsinogen, and we have this site over here. Once we have pH 4, this site is going to be cleaved, and what we're left with is this. And this is pepsin. This is the active form of pepsinogen. So pepsin is the active form. Pepsinogen is the zymogen, or the inactive form. And then once this pepsin is converted, it catalyzes the rest of the conversion. So the pH 4 is only required for the first few, and then the rest of them are catalyzed by pepsin themselves. So what stimulates pepsin? It's quite simple. I think remembering this is not the best way to go. I think the best way to do this is logic. So when do we need pepsin? We need pepsin when we eat, because we need the food to be digested. So the first thing that stimulates pepsin is food ingestion. Okay. We also know from earlier that gastrin stimulates pepsin release, because it also stimulates HCl release, and they're correlated. So food ingestion, food, gastrin, and then ACH. And you might say, well, ACH, that's so random. How am I supposed to remember ACH? And I think this could help. It helps me anyway, so maybe it helps you. So when you think of ACH, think of parasympathetic. I know you don't know the details of parasympathetic, but you do know the basics of parasympathetic. So when you think ACH, think parasympathetic. Parasympathetic system is active when you're eating food, because parasympathetic means you're chilling. And usually when you're chilling, you're having food, you're drinking something. So basically you're chilling, it's parasympathetic. And parasympathetic is ACH. ACH is responsible for parasympathetic. That's how I like to remember. You can remember it whatever way you want. At the end of the day, you just need to remember that acetylcholine, food ingestion, and gastrin stimulate pepsin. And what inhibits pepsin? Easiest thing in the world, somatostatin. I told you it's always an inhibitor. It inhibits, that's its function. So somatostatin inhibits pepsin. Now, is pepsin important? It's not that important, because we have other proteolytic enzymes that break down proteins coming from the pancreas. So pepsin, sure, it helps, but it doesn't make that much of a difference if it's not present. And that's why when you have gastrectomy, which is removal of the stomach, or eclarydia, which means no HCl or pepsin, it doesn't affect the person that much because the pancreas is going to take care of it. That's it for pepsin, and it's really easy. They have a diagram right here, but it's not very important. As you can see, the HCl, it breaks down this protein into this, it denatures it so that it's easier for the pepsin to break the proteins into smaller parts, which are peptides, right? That's it for this slide. If you have any doubts, obviously, you can put it in the chat. Now we have gastric mucus. So I'm going to start with the picture, and I just put this picture in because it's really cool. So you see the green part of the picture are the mucus glands of the stomach. So this is the lumen of the stomach, and these are the glands. These, you can follow my outline. So this is mucus. The pink one is the mucus that's being released, and I just thought it was a really cool picture. So what stimulates the release of mucus? It's acid secretion. So acid, if you have an increase in acid, you have an increase in mucus. And this is really simple. Like, as I said, if you think about it logically, obviously it makes sense, because if you have an increase in acid, you don't want to harm the stomach. You want the stomach to remain okay. You don't want any ulcers or injuries in the stomach. So what do? You try to protect the stomach. And how do you protect the stomach? Through mucus, because this produces mucus and bicarbonate, which neutralizes the acid. You want the stomach to be safe, and that's why when you increase acid, you increase mucus. And histamine and acetylcholine also increase mucus secretion, but you should already know this. And the reason is because of the histamine, gastrin-histamine axis. Do you guys remember this, or did you already forget? The gastrin-histamine axis, which means that if there is an increase in gastrin, there's going to be an increase in acid. Okay? So, and if there's an increase in gastrin, that means there's an increase in histamine. So an increase in histamine also means there's an increase in mucus. Think about it like a math equation. If this equals to this and this equals to this, then this also equals to this. Okay? So if you have acid in the stomach, that means you had the gastrin-histamine axis activated, which means there should be an increase in histamine. And hence an increase in histamine is correlated with an increase in mucus. Similarly for acetylcholine. So just remember that. And obviously, if you have decreased acid, you don't need as much mucus, so the mucus secretion is also going to decrease. Okay, now we have the phases of gastric acid secretion. Before we go to the phases and we're finished with another part of the lecture, this is the last phase of the lecture. It's not that long, but if anyone has any questions from the previous few slides, you can ask now. You can unmute your mic and ask, or you can put it in the chat. Okay, so let's move on to the next phase then. We have integrated regulation of gastric acid secretion. And we have the different phases of gastric acid secretion. So the HCl, or the hydrochloric acid, is activated along with pepsin simultaneously. And increased hydrochloric acid increases mucus secretion. This is everything that we've learned in the previous slides. So that must mean, through simple maths, that HCl, mucus, pepsin are released simultaneously. They're released together so that the mucosal integrity isn't compromised, because we don't want HCl to be released and then after a few minutes you have mucus release, because then in those few minutes the HCl can obviously harm the cells in the stomach. So they're all released together. And now we have the phases. So there's 4 phases. We have the basal phase, which is the base. This is the base, and it's always around this much. So this happens between meals when we don't really need any acid. Because why would we need acid between meals? There's nothing in the stomach. Then we have the cephalic phase, which is 30% of the time. This is a stimulus from the central nervous system, and this happens after we digest the food or during we're digesting the food. So that's cephalic phase, and this increases the acid. We'll learn how it increases the acid in a second, but for now, just know that it increases the acid. Then we have the gastric phase, which is 60%. And this is stimulus from the stomach, and it also increases acid. So cephalic is stimulus from the central nervous system. Gastric is from the stomach, you can tell by the name. And intestinal is from the intestine. And intestinal decreases acid. This is the only one that decreases acid. And the reason it decreases acid is because all of these are in anticipation of food. So basal is between meals, so there's no food in the stomach, but you still have some acid. Cephalic is when you're digesting the food, so you're anticipating food, so you want to increase acid secretion. Gastric is when the food is in the stomach, and obviously you want to increase acid secretion if there's food in the stomach. Intestinal is when digestion is coming to an end. So digestion, the whole process of digestion is ending, which means soon there's going to be nothing left in the stomach. The stomach is going to be empty. So why would you need acid? You don't need acid in the stomach if it's empty. And that's why intestinal stimulus decreases acid production, because it tells the stomach, okay, there's nothing left there, you can stop producing acid. The phases are not discrete, they happen concurrently. So while one part of the stomach, like suppose, let's say, the proximal part may be in the basal or cephalic phase, the distal part could be in the intestinal phase, because the stomach doesn't only have, okay, let me give you this example. Like, let's say you ate food 3 hours ago, okay? That food may be in the distal part of the stomach, right? And this food is mostly digested. It's going to be digested. It's going to be ending. So the distal part of the stomach might be in the intestinal phase. But let's say you just, let's just say you ate 15 minutes ago too. So you ate 3 hours ago, and that food is distal, in the distal part. But you also ate 15 minutes ago, and that food is probably in the proximal part. So the proximal part may be in the cephalic phase right now. So they're not discrete, they're continuous. So one part of the stomach the different parts of the stomach could be in different phases, depending on when you last ate. So basal secretion between meals. Why do we have basal secretion? Why? What's the reason? And the reason is due to the vagal tone. The vagal tone is due to the vagus. The vagus is the 10th cranial nerve, obviously. So the vagus is the most famous and most well-known parasympathetic nerve in the whole body. Oh, wait, no, not parasympathetic, my bad. Damn, that's embarrassing. The vagus, which is the 10th cranial nerve obviously comes from the head, is a very important, it regulates, and it's the main parasympathetic nerve. So the vagus is the main parasympathetic nerve. I was right. Never mind. I had to search that up, but yeah, I was right. It's parasympathetic. I don't know why I got confused. So the vagus is parasympathetic, and parasympathetic means, and usually when you eat food, you're in the parasympathetic nervous system control, because when you're chilling, that's when you eat food, and parasympathetic is chill. So vagal tone is important. And the basal secretion is due to this. So the vagus always has a small connection with the stomach, and that connection is basically what causes the release of the ACL during the basal period. And it can be removed due to a vagotomy, which is removal of the vagus nerve, or atropine, which is an antimuscarinic. So the muscarinic receptors are parasympathetic, but if you have an antimuscarinic, then it would block this pathway. So in either case, you would have no vagal tone, which means no basal secretion, right? So no vagal tone, no vagus, means no basal secretion. So now we talked about why we have a basal secretion. So how is the basal secretion blocked? So let's say we have too much basal secretion. What do we do? So as I said, like, somatostatin is always an inhibitor. As you've seen in this lecture so far, every slide, whenever there's anything next to the word inhibitor, it's always somatostatin. So somatostatin always inhibits. So somatostatin is the main inhibitor for basal acid secretion as well. So the reason for this is obviously it inhibits G cells, which produce gastrin, and gastrin obviously leads to release of pepsin and HCl. So if gastrin is blocked, then so is hydrochloric acid. It also inhibits ECL cells from releasing histamine, which blocks the gastrin-histamine axis, right? So again, the gastrin-histamine axis eventually leads to the formation of hydrochloric acid. So again, this is blocked. This is blocked. It also inhibits parietal cells directly from forming HCl. So the somatostatin inhibits it in 3 ways. So there's 3 different pathways by which it inhibits the release of hydrochloric acid. So then the question arises, wait, so the somatostatin inhibits basal secretion, then why is there basal secretion? Because we know that there is basal secretion because I told you there is basal secretion over here, 10%. Why? So the answer for this is the vagal tone, which is the connection between the vagus and the stomach, minimizes the impact of somatostatin. So it fights with so it's basically the vagus versus the somatostatin, and the vagus is a bit stronger, and that's why you have the 10% release of HCl, but it's a minute amount. It's only 10% of the normal amount. Okay, so now we talked about what basal acid is. Okay, we talked about how to inhibit it. We talked about how the vagus overcomes the inhibition. But what if there's too much basal acid? Then what? What do we do then? So then we have this amazing system called the calcitonin gene-related peptide system, which is this. And this is involved in negative luminal feedback. So if there's too much acid in the stomach during the basal phase, which means when there's no food, this is going to lead to a decrease in the pH, right? Because it's acidic. And if you have this acidification, low pH, it activates enterochromaffin cells. You guys remember enterochromaffin cells? They were the little pink cells at the bottom of the gastric pit. So those enterochromaffin cells will release serotonin. You guys remember serotonin? Enterochromaffin cells release serotonin. Enterochromaffin-like cells release somatostatin, which is the inhibitor. Enterochromaffin cells release serotonin. So serotonin activates sensory neurons to release CGRP, which is a neurotransmitter, calcitonin G-related peptide. What does CGRP do? CGRP stimulates somatostatin. By the way, SST is somatostatin, in case you didn't figure that out. Somatostatin. Right. So CGRP stimulates somatostatin, and somatostatin blocks the G cells, which blocks gastrin release, which blocks the release of HCl. So somatostatin blocks the G cells, and if there's no G cells, I mean there's no gastrin. If there's no gastrin, there's no HCl, because gastrin-histamine axis, guys, remember? So this CGRP is also a very important point from the point of view of examinations. So you should know this. So CGRP is the mechanism that's used when you have too much basal acid. And you can see, like, we thought about 4 different scenarios. What is basal acid? What is basal acid? How is it inhibited? If it's inhibited, then how does it form still? And if there's too much, then what happens? So 4 different scenarios, and for each scenario, we had a different type of regulatory mechanism. So this whole process is very well regulated because we do not want too much or too little, because both of those are harmful. So one last time, if there's too much basal acid, we use CGRP. If we have basal acid, then why is it? Then how do we inhibit it if it's too much? It's through somatostatin. If why do we have basal secretion? Because of the vagal tone, right? So everything is covered. So now we're done with the basal phase, which was the first phase here. This is the first phase, the basal. And now we go to cephalic. The cephalic phase is activated by the central nervous system. So the central nervous system is responsible for sight, smell, and taste, right? So all of those things are controlled by the central nervous system. So all of those things lead to an increased vagal activation. So anytime we see food, anytime we smell food, anytime we taste food, we have an increased vagal activation, which leads to an increase in acetylcholine and parietal cell secretion. And the reason for this, obviously, is I told you to remember it this way. Vagus is parasympathetic. Parasympathetic means you're chilling, right? And when you're chilling, you're eating food. And if you're eating food, then what do you need? You need the things to digest the food. And what do we need to digest food? We need parietal cell secretion, which is HCl, right? So the vagal activation increases parietal cell secretion. And then you remember in the start of the lecture, I said GRP. GRP stimulates gastrin release, and then gastrin does the whole gastrin-histamine axis. So GRP is gastrin-releasing peptide. And this is also activated by the vagal activation during the cephalic phase. So all of this is controlled by the central nervous system. So in this case, we talked about the stimulatory factors for acid secretion, and we talked about the inhibitory factors. So in this case, the stimulatory factors win, and you have more acid secretion. So cephalic phase is CNS, and how I remember this is C. Okay. So CNS, sight, smell, taste of food, vagal activation increased, which increases parietal cell secretion. You can obviously, if you have any doubt at any time, you can put it in the chat. By the way, the next lecture is much easier than this one, and it's so short. Like you could finish it in 20 minutes. So I'm sorry this is taking a little bit time, but we're almost done with this lecture as well. Gastric phase. So this is the third phase. The first one was the basal one, the second one was the cephalic. Now we have the gastric phase. And this is activated when food enters the stomach. The cephalic phase is when like the food enters your mouth or you see the food. This is when food enters the stomach. So let's say this is the stomach and this is some food. When it enters the food, when the food enters, obviously the stomach is going to distend. It's going to become bigger. And this distension activates enterochromaffin cells to release serotonin. And serotonin activates the vagal afferent nerves and also the vagal afferent nerves. Okay, you can forget that. Just remember, serotonin activates the vagal reflex. Okay. So because the stomach is getting bigger, it's distending, you have the activation of enterochromaffin cells. Now you might be like, okay, why is the enterochromaffin cell being activated? And this is because enterochromaffin cells have mechanoreceptors, which means that basically when they sense any pressure, they get activated. So they're activated, they release serotonin, and serotonin causes the vagal reflex to be active. And literally the last slide, what did we learn? The vagus releases ACH and stimulates parietal cells. So it does the same thing here. Yani it's not going to change. It does the same thing, ACH and parietal cells. It also activates the ECL cells and G cells. So as that, like literally, if you remember this, you don't need to remember anything else. The vagus is parasympathetic. Parasympathetic means chill. Chill means food. And what do we need for food? We need all the other things that are responsible for food digestion, which includes parietal cells, HCl, ECL cells produce histamine, which is for the gastrin-histamine axis, and G cells produce gastrin, which is also for the gastrin-histamine axis. That's why the vagus activates them. Now we have food entering the stomach. Let's say we have food here. This is broken down by pepsin, and this is through a process called protein hydrolysis, right? So when we have this protein hydrolysis, it's detected by G cells, and they release gastrin to further increase the speed of hydrolysis, so we can have more HCl. So the peptides produced by protein hydrolysis are detected by G cells. Now we have food buffers in the lumen. So we have this food, right? It acts as a buffer. So now we have HCl. Let's say we have HCl over here. The food is basically going to make the stomach less acidic than if it was not there. So this food is going to make the stomach less acidic. It reduces the effectivity of the acid. So the stomach reduces how good the acid is working, basically. You can frame it, if you want to put it really simplistically, it reduces how good the acid is working. And this increases the pH, because when you have decreased acidity, you have increased pH, right? And you remember this pathway? I'm going to show you the pathway again, so you remember it. If you have increased acid or low pH, you have activation of EC cells, and EC cells release serotonin. Serotonin activates calcitonin gene-related peptide, And calcitonin-gene-related peptide stimulates somatostatin. Somatostatin is an inhibitor. It always does that, and that's why it inhibits G cells. Remember this, low pH. Now, what do we have here? We have an increased pH. So the CGRP pathway is going to be, like the CGRP pathway is not going to work. So what is the end result? That means there's no somatostatin, which means there's no inhibition. That means there's going to be a lot of gastrin produced. And when we have gastrin, we have the gastrin-histamine axis, and we have HCl. And that's why when there's food in the stomach, it reduces the effectivity of the acid, which decreases the pH, but this in turn, which decreases the acidity. When we have food in the stomach, it makes the acid less useful, which means there's an increase in the pH because the acidity is decreasing. And then we have the inhibition of this calcitonin-gene-related peptide pathway, which means there's an increase in gastrin because somatostatin is inhibited along with this pathway, because this pathway leads to somatostatin. So the net effect is more acid secretion. So this one is, I can tell it's a bit confusing. There's these 3 points: distension, the protein hydrolysis, the P hydrolysis, and the food buffers. Those are the 3 points you need to remember. I need to remember the mechanisms. So we're done with 3 phases. We have one last phase left, and that's the intestinal phase. During gastric emptying, which is basically when the food is leaving the stomach and going to the duodenum, which is the first part of the small intestine, the acid returns to basal levels. And why is this? So basically, whatever we did, just opposite. So remember how in the cephalic phase, C for C, we have the CNS. And the CNS is sensory, obviously. So when the food is leaving the stomach, you have decreased sensory cues, or when the food is finishing, which means there's a reduction in food-induced cephalic phase. Cephalic because C for C, remember? And we have reduction in distension-induced vagovagal reflex. So remember, the vagovagal reflex is due to the distension, and that's sensed by the enterochromaffin cells. So the vagovagal reflex is also going to stop, which means it will no longer lead to an increase in acid like it did earlier, because we need acid if we have food. So that's one thing that happens when you have gastric emptying. It's the opposite. You've done all of this, you're just flipping it around now. The peptides leave the stomach. Remember how I said if we have peptides produced by protein hydrolysis, G cells are going to sense it. G cells are going to be like, oh, okay, there's a peptide here. So when the peptides leave the stomach, the G cells are no longer going to be stimulated by them. And that's why you're going to have a decrease in gastrin. Also, when the food, which was present in the stomach and was buffering, right? It was a buffer. It was reducing how effective the hydrochloric acid is. It's going to leave the stomach. It's going to leave. That means the hydrochloric acid is once again going to be more effective. And hence, you're not going to have the buffering of, the buffering obviously leads to an increase in the pH. That means it will inhibit the CGRP pathway. But if you have a normal pH, because there's no more buffer, then you're not going to inhibit the CGRP pathway. And the CGRP pathway is going to lead to somatostatin production, which is an inhibitor of acid. So that's why when you have gastric emptying, you just flip everything around, and that's what happens. You have a reduced acid secretion. Also, one last thing. I already mentioned this in the first slide, but intestinal phase, which is the 4th phase and the last one, is the only one that leads to a decrease in acid. All the other ones lead to an increase in acid, okay? Because this is the only phase where the food is leaving. In all the other phases, the food is going to come into the stomach, which means it needs to prepare, which means it needs to increase acid. In this one, it's leaving, so it doesn't need acid anymore. That's why you decrease the acid. Okay, this is the last one. Look, I promise this is the last slide. Look, you can see the view, which means last slide. But before that, do you have any doubts? I'm going to give you 30 seconds to type in anything.
Yeah, someone asked to recap the gastric phase.
Gastric phase. Okay. Okay. Bismillah. I know this is a bit tough. I swear it's tough for me too the first time. But after you understand it, it's literally the easiest thing in the world. The first thing we're going to talk about is distension. Distension is sensed by EC cells. EC means enterochromaffin. How does it sense the distension? It's because it has mechanoreceptors, which means it can feel the pressure. When the food enters the stomach, it's going to make the stomach expand and it's going to put pressure on the stomach. So that's why we have the distension, and this activates EC cells. EC cells release only one thing, and that one thing is serotonin. Serotonin is going to activate the vagovagal reflex, which is basically the afferent and the efferent. You don't need to know that. Just remember vagovagal reflex. Now, here's the thing. It's hard to remember what the vagovagal reflex does. Like, if you were to remember it in a vacuum, it's hard. But the way I think of it is vagovagal is equal to vagus, right? And vagus is parasympathetic. Parasympathetic is the chill part of the nervous system. And when you're chilling, you're eating, okay? And when you're eating, what do you need when you're eating? You need something to break the food down. And that something is HCl by the parietal cells, histamine by the ECL cells for the gastrin-histamine axis, and gastrin by the G cells, okay? So that's how the vagovagal reflex leads to the activation of all of these. It leads to the release of hydrochloric acid, histamine, and gastrin. Okay, that's the first part. That's distension. Distension is out of the way. Okay, we did it. Khalas. The second thing is peptides. So, you know, we have proteins when we eat, and the proteins are broken down by pepsin, right? The pepsin breaks it down into peptides. And these peptides are sensed by G cells. So the peptides and G cells have a connection. When the G cells identifies that there's peptides in the stomach, it's going to increase gastrin. And what happens when you have an increase in gastrin? Gastrin-histamine axis increases, which means you have more gastrin and more hydrochloric acid. So that's two things out of the way. We did peptide and we did distension. The third thing is buffer. Now, this may be a little bit difficult, so I'll explain it like fully again. This is the stomach. Just bear with my very anatomical drawing. This is food in the stomach. The food is going to reduce how effective the HCl is. So let's say the HCl is 10 out of 10 before the food comes, and after the food is 5 out of 10. So now you have reduced HCl effectiveness, which means the pH is not going to be as low as it was earlier. Earlier it was low, but now no more. Now it's more. You have more pH because it's less effective, right? So now this pH is increased. And I'm going to take you back to this slide so that you can completely understand this. If there is a decrease in pH, then you have EC cells activated and serotonin is released. Serotonin leads to the release of this neurotransmitter called calcitonin gene-related peptide. Calcitonin gene-related peptide leads to a release of somatostatin, and somatostatin inhibits gastrin. This is the case when we have low pH. But in this case, what do we have? We have a high pH, right? Because the food is making the HCl less effective. So this whole pathway cancelled. It's gone, okay? Which means no CGRP, no somatostatin, no inhibiting gastrin, which means gastrin is going to be present. It's not going to be inhibited. And if we have gastrin, that means we have hydrochloric acid through the gastrin-histamine axis. So these three things work together and we have more acid. Is that clear? If you have a problem, I'll repeat again because I understand how stressful, like when I was making the PAL session, the first few times I had to go over it again and again. But when you go over it, then it's super simple. Like it's just common sense then. So if you still have a problem, tell me. I don't mind repeating again. And we're on the last slide. Look, this is the review, and I'm just going to go over this after I finish the slide, and then we're going to take a break. And the next lecture is, I promise, it's so simple. It's so simple. Okay, intestinal phase is the last phase, the fourth phase, and this is the only one that decreases acid, right? Because now we don't need it anymore because the food is emptied into the intestine. There's nothing in the stomach. So the chyme, or chyme, I don't know how you pronounce it, chyme, I guess, is basically food that has been mixed in the stomach, and it's mixed with all the gastric acid and all of the stuff. It's small particles of food. When this enters the duodenum, it can stop gastric secretion. And it does this through three pathways. Actually, there's only two mentioned here, so we'll only mention two. So the first thing is the chyme is obviously acidified. And this means the reason it's acidified is because obviously it was mixed with HCl, right? And when you have this acidified chyme, you have these cells called S cells. S for S. S cells release secretin. Remember it this way, S for S. Then you have fatty acids in the chyme, right? Because obviously, when you have the lipases that are produced in the stomach, they break down the triglycerides into fatty acids, right? The fatty acids activate the I cells, and this leads to a release of CCK. Look, I'm sorry that there's so many names, but this is not my fault. This is the fault of whoever made medicine, right? It's not my fault. S for S, secretin. I cells, CCK. Okay? So secretin and CCK are released when you have chyme because chyme is acidified and it also has fatty acids. So when CCK and secretin are released by these cells, they mediate the efferent vagal pathway. Okay? And this is not the vagovagal. Okay? I told you the vagovagal is the one that eventually leads to chill, right? And chill means food. And food means you have an increase in whatever you need for food, which means an increase in HCl, gastric, G cells stimulated, and ECL stimulated. But this is efferent vagal pathway, not vagovagal. Vagovagal is the chill one. The efferent vagal pathway leads to an increase in somatostatin, which is the bad guy, the inhibitor. He doesn't want other people to be happy. He wants to inhibit everything. So SST inhibits, right? Somatostatin. SST is somatostatin, don't forget. So the efferent vagal pathway is stimulated by secretin and CCK, which are released by the S cells and the I cells, respectively. Okay, so they're also called enterogastrones. And this is not very important, but just remember that secretin and CCK are enterogastrone. They're pharmacological, which means theoretically, this makes sense. Like we're studying about this, but in actual, like in real life, when the body is digesting food, it doesn't actually play that big of a role unless they're higher than physiologically than they should be. Like if these are normal, then they're not going to make that much of a difference. But if they're elevated for some reason, then they're going to make a difference. So secretin and CCK lead to activation of the efferent vagal pathway, which leads to an increase in somatostatin. Efferent vagal, I promise, I swear, if any of you guys make a mistake in this, I'm going to be so pissed. Efferent vagal is not the same as vagovagal. Vagovagal is different from efferent vagal. Vagovagal is the good guy that leads to chill and food. Efferent vagal is the bad guy that leads to somatostatin. Just remember this, please. Like, if this makes you remember it, then it's worth it. Efferent vagal, somatostatin. Vagovagal is the cool guy, leads to food, which means everything that we need for food, which means we will have HCl, stimulation of G cells, and stimulation of ECL. Okay, we're done with this lecture. Alhamdulillah, we're just going to review this real quick. I'll not explain in detail, just review. So we have this graph, and as we know, Dr. Abdul Jabbar loves to bring graphs. It's his favorite thing in the whole world because most people get them wrong. But all you need to know is how to read the graph. And luckily for us, most of it is labeled here. So we see the first part. This is when we're eating or before the meal. This is before the meal is basal. So this is the base, right? Now we're eating. So we see the pH of contents increases. Increased pH. Why is this? It's because the food is buffering, right? I told you the food is buffering. And if the food is buffering, that means we have an increase in pH, which means there will be an increased HCl release because the CGRP pathway that we talked about is inhibited now. It's stopped. And this is why we're eating, obviously. And then you also see the rate of acid secretion, which is this one. It's also increasing as the food is increasing because it's buffering the food. Now, the food is going out. Like this is the volume in the stomach as well. It's decreasing. That means the food is being digested. So obviously the pH is going to decrease. And this is because the buffering is no longer as useful as it was before. Like earlier, the buffering was a lot. Now it's a little. That's why it's decreasing. And that's why you're also going to see the acid decreasing. The rate of the acid secretion is going to be inhibited. And that's all this little graph is going to show you. Don't mess it up because it's super simple. Okay, here's the whole graph for reference. Okay, it's super simple. Like just read the graph and you're good to go. If you need me to explain the graph again, tell me in the chat so I can explain it. Now we'll move to the next graph. This was obviously while the food was being eaten. This is after the food is eaten. So you see here, the volume in the stomach is decreasing. If the volume in the stomach is decreasing, that means there's less food. If there's less food, then the buffering is going to be less useful. If the buffering is less useful, the pH is not going to be high. It's going to be low because the ACL is not affected when there's no buffer. If there's low pH, then we have the CGRP pathway. Remember this pathway? If you have low pH, then this leads to the CGRP pathway, which increases somatostatin, and somatostatin is an inhibitor. So this is going to inhibit acid secretion, and that's why it's going to get lower. And that's what this graph is showing you. So these are two really simple graphs. You don't even need to explain them because you paid attention in the lecture. This is easy. So if anyone needs me to explain them again, though, it's fine because I know graphs can be tricky. But this is, alhamdulillah, like it's not as bad as other ones. So we're done with this lecture. I believe Dr. Abdul Jabbar said he will not test you on the clinical part, which is this. So I'm not really going to go over it. Aslan, there isn't much to go over. Like it's literally just increase, decrease, and you need to remember. But it's like he does not test you on clinical stuff in first year, obviously. So this is probably not going to come. If it does come, appeal it, and if that doesn't work, that's like it's just a few letters. Just learn it. So now one last thing. I promise. Look. This is the next lecture, and we're done with this lecture. Protective factors for mucosa and damaging factors. So what protects the mucosa? The first thing we said is the mucus itself, released by the mucosal cells. We have the bicarbonate, which is released also by the mucosal cells, and this neutralizes the acid and prevents it from hurting the stomach. We have prostaglandins. The prostaglandins, like it's very easy how it protects. So basically, it increases the blood flow to the stomach because it's a vasodilator. It leads to an increase in the vessel's size and more blood flows. And if you have more blood, obviously the waste materials are going to leave quicker, which means there's going to be less damage to the gastric wall. Obviously, an increase in mucosal blood flow for the same reason as prostaglandins also helps, and growth factors obviously also help because they're growth factors. So damaging factors, H+, because H+ is acidic, and acidic is bad. Pepsin, obviously, because of the same reason. H. pylori, which is a bacteria, you don't need to know about it. NSAIDs, because they're proton pump inhibitors, okay? And, you don't need to know this, just forget this because this is too much. Stress. The stress is bad for you in any way, like it's bad for your stomach, it's bad for you as a whole, that's why you don't stress. Smoking, obviously, and alcohol, all of these will damage the mucosa. And that's it. We're done with the first lecture. So if you have any doubts, tell me, and then we're going to take a break. So yeah, tell me if you have any doubts. Any doubts? Okay then. We can take a break now. Is that okay?
Should we take it for 10 minutes?
Yeah, sure. 10 minutes.
Okay.
We can come back at 9:45.
Okay.
Okay. Okay. Now we have the second lecture. Super simple. Much easier than the first one, I promise. And it's not going to take more than 40 minutes, inshallah. So why do we gastric motility? First of all, why do we have gastric motility? Gastric motility is basically the movement of food through the stomach. Why? First of all, is a combination of food with little change in pressure. The pressure in the stomach is always constant, so it never changes. How? How is that possible? Because let's say this is the stomach and this is like normal. Then we add food in it, and I told you earlier that it distends. So how does it not increase the pressure? And the reason for this is because we have a proximal gastric tone in the stomach. And this proximal gastric tone is relaxed when they have food in the stomach, which means that it's not going to be rigid, it's not going to push back. So this is going to lead to a decrease in the pressure. And another function why we need gastric motility is for the contractile activity to mix food. and to mix food with the gastric secretions, and also to reduce the size of the food into small particles. And the last one is because we need the motility to empty the food inside the stomach into the duodenum, or the chyme into the duodenum. I swear this lecture is super simple. It's like a high school lecture. Okay, we have two divisions in the stomach according to what we have to study today. Obviously, we have different divisions, but for today, we only need proximal and distal. This red line separates proximal and distal, this one. The proximal stomach has little contractile activity, so no major contractile activity. It has no slow waves, and I'll tell you what slow waves are later. Just remember that the proximal stomach does not have them. And it mainly stores food. So when you eat and the food is stored in your stomach, most of it is stored in the proximal stomach, in the fundus. I'm sure you guys already took the anatomy of the stomach. So the fundus is where most of the food is stored. And how can it store this food? The first thing, as we said earlier, when the tone decreases, the proximal gastric tone. Second thing, the muscular layer in the proximal stomach is super thin, which allows it to be flexible, and hence it can expand to store food. And the third thing, and the most important pathway, is receptive relaxation. Let me write it down. Receptive relaxation. Right? So what is receptive relaxation? So remember the vagovagal reflex, right? I swear, if you guys don't remember it after all of this, I'm actually going to be like, I'm going to be so sad. Vagovagal is the cool vagus, and vagus is parasympathetic, and parasympathetic is chill, and chill means food. So the vagovagal reflex is going to do everything that makes it possible for food to be digested. So the vagovagal reflex receptors in the pharynx, which is a part of the digestive system, activate due to chemical, thermal, and mechanical stimulation. So the chemical stimulation could be the taste, something like bitter, something sweet. Thermal could be temperature, cold water, hot coffee. Mechanical stimulation, the food that's being chewed. So this activates the vagovagal reflex. Then we have distension. I already told you the EC cells, or the enterochromaffin cells, have mechanoreceptors. And when the food is being chewed, it's going to put pressure on these receptors and going to allow them to expand. And when they expand, the EC cells are going to release serotonin, right? Similar to what happened in the stomach, serotonin. And serotonin, this time, is not going to activate the nervous system to release this peptide called CGRP. That was what happened last time. This time, it's going to activate the enteric nervous system. The enteric nervous system is a part of the peripheral nervous system that is only focused on the stomach. So the enteric nervous system is going to lead to a release of VIP, vasointestinal peptide, and NO, nitric oxide. Both of these are relaxants. I don't know if you guys had this lecture, but in foundation, we had this lecture where nitric oxide's pathway was explained to us. So basically, it leads to an increase in cGMP, and then eventually it leads to relaxation of the muscle. So the same thing happens here. When when the serotonin is released due to the mechanoreceptors activating the enterochromaffin cells, you have stimulation of the enteric nervous system. This leads to release of VIP and NO. NO uses the cGMP pathway, and vasointestinal peptide is obviously, like it's in the name, it causes vasodilation, so dilation of the muscle. So both of these things together lead to relaxation and dilation of the muscle. So the vagovagal reflex and the enteric nervous system together relax the upper part of the stomach, allowing for more food to be stored there. The proximal stomach has slow tonic contraction and relaxation. Relaxation is when it's accommodating food. So if you're eating, like if there's food, it's going to relax, right? To become bigger, to allow more food to enter. And when it's trying to empty, when it's trying to move the food into the pylorus from the fundus, that's when it's going to contract. But this contraction is slow tonic contraction. It's very distensible. And the reason that it's distensible, obviously, is because it has a very thin wall. It has the receptive relaxation thing. And then it also has the relaxation of the proximal gastric tone. And I told you there's a proximal stomach and a distal stomach, and they're separated by the red line. So what separates them? Is it like random? Like did someone just pick up a ruler and say, okay, this is around half, so we'll do this? No, that's not how it happened. The reason is the basis of division is the contractile activity. And the contractile activity is given to the stomach by the pacemaker region, which is circled over here, the pacemaker region. So the pacemaker region, let's discuss it first, and then I'll get back to how it's divided. It sets a rate of BER, which is basal electrical rhythm. So basal electrical rhythm is an impulse that's given out by the pacemaker region, and it eventually it transforms into a peristaltic movement. Peristaltic movement is a contractile movement, and this BER spreads towards the antral contractile area, which means this way, right? Because antrum is this way, so it moves this way, distally, and produces propulsive contraction. And this contraction obviously allows the food to move into the next part of the GIT, which is the duodenum. The BER is 3 cycles per minute, and this is a set rate, similar to how the AV node and the SA node have rates, because you do gastric CVP. So the BER has its own rate, 3 cycles per minute. So the basis of division now is the pacemaker region gives the BER distally, right? So the areas which get the BER, which are influenced by the pacemaker region, are the distal stomach. And the areas that are not influenced by the pacemaker region, that just have tonic contraction through the vagal tone, is the proximal region. So the proximal region has no, the pacemaker region has no influence on the proximal zone, right? And this means that they have, by the way, do you remember that the proximal zone has no slow waves? And this is why, because the pacemaker region does not have any effect. So there's no slow waves here. And I'll tell you what slow waves are, I promise. But the distal one does have slow waves, because the pacemaker region, it influences the distal area. Fundus empties via contraction of muscle layers to the pylorus. So the fundus, when it wants to empty, it moves the food to the pylorus. And how does it do this? It's through the muscles. So it has 3 layers. I'm sure you guys already took this in histo: outer longitudinal, middle circular, and inner oblique, right? And as you can see, you see this underdeveloped. What does that mean? That means that these muscles are thin, and this is why the fundus can hold so much food, because these muscles are thin and they can expand easily without much opposition. Okay, that's it. Distal stomach. Now we're done with proximal. We're coming to distal. It has slow waves. So proximal, P for proximal, D for distal. It has slow waves. Present. Slow waves here? No, not present. Okay. It has strong peristaltic contraction, and the reason for this is because it's influenced by the pacemaker region, by the BER, basal electrical rate. So the peristaltic contraction helps in mixing and moving the food into the next part of the duodenum, which is the duodenum. It's slow distensibility because it's not responsible for storing food. It does not need to be flexible and distensible to store food. Like, its only function is to mix the food and grind it into small pieces. And that's why it's rigid, because since the stomach wall is rigid, it's not going to expand. It's going to instead stay where it is and compress the food when it comes in. And this compression obviously leads to grinding the food and making it into small pieces. So its only function is gastric mixing, mixing the food with the HCl, with the pepsin, with the other enzymes, and emptying into the duodenum. It's under neuroregulation via the enteric nervous system and the vagal tone, right? So a very important rule in all of GIT is the vagus is a very important part of GIT, because the parasympathetic is very closely linked to the GIT, and that's how it's controlled. So vagal, remember vagal everywhere. And remember the difference between vagal and afferent vagal, please. Slow and contractile waves. Now, I've been saying what are slow waves, what are contractile waves. I've said a lot of words, but what are they? So I'll start from this area. What is the difference between slow and contractile waves? So the pacemaker region of the stomach is going to be an impulse, and it's going to be developed into a slow wave. When the slow wave reaches a threshold, it triggers the opening of voltage-gated calcium channels. So the calcium channels open, leading to an influx of calcium ions into the smooth muscle. And this calcium influx initiates muscle contractions, because when you have calcium inside the cell, it's going to contract, resulting in the propagation of peristaltic waves through the stomach. So the pacemaker leads to slow waves. And then the slow waves overcome a threshold, leads to the influx of calcium coming into the cell. If this is the cell, the calcium is coming into it. And then the calcium in the cell leads to further contraction. And this further contraction is peristaltic contraction. Peristaltic contraction or contractile waves. The contractile waves are much stronger than the slow waves, but they come from the slow waves, basically. The slow waves are basically impulses. Like if you think about it, like an impulse comes to a muscle and the muscle contracts. The same way the slow wave is an impulse for further contraction of the muscle. Okay. Now, slow and contractile waves move from the pacemaker region to the distal stomach. They increase in speed and strength. So, as I said, the antrum is where they're going because only the distal stomach has the slow and contractile waves because that's the only region that's influenced by the pacemaker region. The other regions are not influenced by the pacemaker. So when they move towards the antrum, they increase in speed and strength. Okay, super simple. And then the pacemaker region produces electrical impulses that are converted to peristaltic contractions eventually. Easy. This is a super nice diagram. As I said, GIT can be explained on diagrams. So we have these equidistant sensors, which is 1, 2, 3, 4, 5, and they're measuring slow waves and contractions. Let's say it's over here. This is the pacemaker region. It's not going to go distal because this is, it's not going to go proximal. This is proximal and this is distal, right? It's not going to go proximal because I said the pacemaker only influences distal. So proximal, there's no slow waves. Look, 0. No contraction, 0. Because the only contraction in the proximal is the vagal tone because the pacemaker does not affect it. Now, as we move to the other sensors that are placed distally, and the distal obviously is the region that's influenced by the pacemaker, you see that the slow waves, they're starting. The slow waves are starting, like, look at them. And then, as you see, once you move more distal, once you move to the antrum, the slow waves, their amplitude remains the same, but they get longer. Look, this is how long it is in the start, and then look at how much longer it is later. And the contractions increase because the slow waves, obviously, they're becoming longer. The contraction is increasing towards the antrum, and that's what I said over here. Increase in speed and strength. Look, this is the contraction in the middle, and then as you get distal, it gets longer, and then in the antrum is the longest, right? So that's basically what I was trying to show you. The red line is basically just showing you, like, at this point, this is how much contraction there is. So the contraction increases as you go distally, and that's what it's trying to show you here. Now, the effect of peristaltic contraction on gastric content. So what happens now that, okay, we understand we have peristaltic contraction, but what exactly happens if you have peristaltic contraction? And the answer to that is in this slide. It's very simple, like it's literally just logic. So the first thing I need to tell you is the pyloric. You guys know the pyloric opening. It's guarded by the pyloric sphincter. It's between the large part of the stomach called the pylorus and the duodenum, right? So it's transient. And transient means it doesn't stay open all the time. It opens and closes. And chyme is bidirectional. That means the mixed up food can move in both directions. It can move from the stomach to the duodenum and it can move backwards. So the large chyme components are forced back towards the fundus with peristaltic movements. The contractions are so strong that the pylorus and antrum are occluded, they close, and the contents are pushed backwards towards the fundus. So let's say we have this food, right? And it's broken down into these pieces. These pieces try to cross the pylorus and go into the duodenum, but they're too big. They can't be digested properly in the intestine. So they need to go back to the fundus where they came from and become smaller. Once they become smaller, then they'll be allowed to go through the pylorus into the duodenum. And how do we know they're small enough? Basically, if they're less than 2 millimeters, that's when you know it's small enough to go through the pylorus and into the duodenum. If it's bigger than that, it's going to go back. It's going to go through a process called retropulsion. I did write it down somewhere here. Oh, retropulsion. Here we go. Retropulsion. They're going to go back to the fundus where they came from. They're going to be grinded more. They're going to become smaller until they're small enough to pass through the duodenum. Super simple, like literally something a high school student can understand. If you have a smaller component that's less than 2 millimeters, which is ideal, almost liquid, they can pass through the pylorus easily because even if the pylorus is constricted, it's small enough to go through. Okay, super simple. If anyone has a problem, by the way, please tell me because I don't expect anyone to have a problem, but still, if anyone does, more than happy to help. So again, a diagram. So these are the small ones. The blue is small, the red are the large ones. The blue ones can easily cross. You can see they're going through this way to the duodenum, but the red ones, because of the constriction, they can't go through because they're too large to go through the constriction. So they're going to be forced backwards into the fundus. They're going to undergo the mixing and grinding again until they become small enough, like the blue ones, and then they can pass through. Okay, so now we have the regulation of gastric motility. So there's two phases, there's two main divisions that we need to know. One is postprandial. Postprandial means after meals. Okay, and the other one is interdigestive, which means basically the fasting period when you're not eating. So when you're fasting, the gastric motility is maintained via the migrating motility complex, the MMC. But during meals and after meals, we have 4 phases, and the names are the same as they were earlier. Remember, during the previous lecture, we had gastric acid secretion, we had a basal, cephalic, gastric, intestinal. Well, here we have the same thing, cephalic, gastric, intestinal. So now we eat cephalic when we see the food, or when the food is in our throat and it's coming towards the stomach. We have the stimulus from oropharynx mechanoreceptors. Mechanoreceptors are obviously those that can sense pressure, and they're in the oropharynx, which is the pharynx, obviously. It leads to receptive relaxation, VIP and NO. Remember receptive relaxation in the proximal stomach, VIP and NO? Well, it's activated by oropharyngeal mechanoreceptors, and this is the cephalic phase. Super easy, right? If anyone has a doubt, please tell me. Second, we have the gastric phase. This is from stomach mechanoreceptors. So it's still a mechanoreceptor, but it's not oropharyngeal. This time it's in the stomach. And this time, the mechanoreceptors, obviously, they're activated and they lead to mixing and gastric emptying. So you have mixing of the food and gastric emptying because the food is already in the stomach. The cephalic phase made sure that the fundus could accommodate the food. The food is in the stomach. Now it's going to be mixed and then eventually moved on to the duodenum. Then we have the intestinal phase, and this is the only phase that leads to a decrease in the stomach function, basically. Like these ones are obviously preparing for the food to come in, so you're going to have increased motility, but now it's going to be like, okay, the motility, the stomach emptying into duodenum can decrease because the food is already in the duodenum. There's nothing to empty, like you don't need to stress yourself, just calm down. So this time it's intestinal mechanoreceptors. So notice how it's mechanoreceptors, like keep that in mind because it's easier to remember if you have an association in your head. So mechanoreceptors, very important. Postprandial phases are not discrete, they're concurrent. That means the same as earlier. The phases are not happening one by one. Different parts of the stomach, different phases can be happening. All right? If you have a doubt, please stop me. So that's it for the postprandial, which is after meals. Now we have interdigestive, which is the fasting. And now we have the migrating motility complex. And honestly, this is very important. I remember for our exam, we had a few questions on this, and also it's generally asked a lot. So it's only 1 or 2 slides as well, so it's really simple. If you pay attention now, it could save you. The resting tone of the stomach, do you remember the tone of the stomach, the proximal gastric tone? It's due to the vagus, vagal cholinergic pathway. Cholinergic is basically to do with the parasympathetic, okay? Vagal pathway. The vagus causes the resting tone of the stomach, remember that. During the interdigestive period, which is the fasting period, we have a migrating motility complex every 90 to 120 minutes. So usually you would expect the stomach in this time to just like be just stay still. Like there's nothing for it to do. There's nothing. Why would it contract? Why would it do anything? Like there's nothing in the stomach. But no, there's a migrating motility complex every 3 to 4 hours if you're fast. Not 3 to 4 hours, sorry, like an hour and a half to 2 hours that interrupts you. And what is the migrating motility complex? I've been saying it, but I haven't explained this. So it's a pattern of motor activity beginning at the pacemaker region throughout the small intestine. So it's some motor activity, which means some contraction, which is beginning at the pacemaker region and then moving throughout the small intestine. Simple. It has 3 phases, the phase 1, the phase 2, and the phase 3. The phase 1 is around 50% of it. There's no contractile activity apart from the tone. So in phase 1, all you have is the vagal tone. That's literally all you have. Okay. Phase 2 is the rest of the 40%. It's erratic, non-propulsive rhythmic contractions. So what does that mean? Let's break it down into smaller words. Erratic means non-rhythmic. sorry, erratic means like with a rhythm, but no specific time. Like it's not like, okay, and then you wait 10 seconds, another. No, it's erratic. It's like one, 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 whenever. Non-propulsive, which means it does not propel the food into the duodenum. Like the peristaltic constrictions are propulsive. They propel the food from the pylorus into the duodenum. This is non-propulsive. Rhythmic contractions, which means they're of the same rhythm. And then phase 3 is the last 10%, and this is intense peristaltic activity for 10 minutes, okay? And when it's peristaltic, that means it's propulsive. That's a landmark of peristaltic movement. The frequency can reach 3 cycles per minute in the stomach, which means 3 intense peristaltic activities per minute in the stomach and 12 in the duodenum, okay? That's phase 3. So phase 1, 2, and 3. And there's a beautiful diagram in the next slide, which makes it so much simpler, and I'll go over it. It's associated with motilin secretion. Motilin is associated with MMC. This is a question that has come before, and it's a very easy question. It's motilin is associated with MMC. What is motilin? It's an enzyme. And we'll talk about it a bit more in the next slide. Okay, now MMC, 70% of them are in the antrum and 30% are in the duodenum, but 0% enter the colon. So the antrum, which is in the stomach, has 70% of the MMCs. 30% of them reach the duodenum. Actually, the MMCs in the stomach MMCs are different from the duodenal MMCs. Like they're different. They come from different ways, but both of them do have MMCs, and they're working the exact same way. However, their origin is different. So 70% are in the antrum, 30% in the duodenum, but 0% enter the colon, which means there's never going to be an MMC in the colon. No, impossible. Okay, in the antrum, which is a part of the stomach, and the duodenum, it's possible. Okay, what is this? I need to erase this, but this diagram is going to make it so much easier for you guys, hopefully, and you'll see how easy it's going to be. So let's talk about it. Okay, this is phase 1, phase 2, and phase 3. Phase 1, remember, is vagal tone. Vagal tone is consistent throughout the stomach, and that's why you see this. All of them are consistent, same length, same time, right? Just as we go down, they're the same frequency and the same amplitude. They're the same because the vagal tone is throughout. Phase 2 is rhythmic contractions that are non-propulsive, right? So you see them happening in the antrum, you see them happening in the duodenum, and then you see them happening in the jejunum, kind of, not really, just a bit, and then they stop before the distal jejunum, right? So the phase 2 is happening in the stomach, but it's not going towards the end of the small intestine. Then we have peristaltic, right? Which is happening in the antrum, it's happening in the duodenum, it's happening in the proximal jejunum, it's happening in the distal jejunum. But as soon as you reach ileum, which is large intestine, it stops. Because we said large intestines do not have any MMCs. It stops before it can reach large intestine, but it's all the way through to the end of the small intestine. The second phase, the rhythmic one, also similarly until the small intestine. The first phase is throughout the stomach, because throughout the GIT, in fact, because it's the vagal tone. So the pathway in a bit, it's slightly more in detail, but we're almost done with the lecture, so just bear with me. It's motilin activates the afferent fibers, which initiates the vagal reflex that brings about change in the gastric musculature. You actually just like scratch that, because this is not even confirmed, by the way. This is just what scientists think bring about the MMC, but actually no one knows exactly to the dot what brings it about. So just remember that the motilin leads to a change in gastric musculature, and that's why you have this MMC, and it does it through the vagal reflex. I mean, even that is optional. Just remember that it changes the gastric musculature. Motilin levels increase during interdigestive periods because the MMCs happen when we don't eat. It happens when we're fasting. Please remember this. MMC is during fasting or interdigestive period. Okay, it does not happen when you eat. It decreases postprandial. So motilin increases when you don't eat, and that's why you have the MMCs, because motilin is linked with MMC. And then it decreases after you eat. So then there's no MMCs after you eat because there's no motilin. Motilin release from duodenum and how it affects the stomach musculature are not exactly known. So we don't know the pathway. We don't even know what happens. We just know that something happens. And that's why we have MMCs. When are motilin levels maximum? This also, I think, is a very important question that can come. I get it's not super clinically relevant or like very important, but this is something that I like to ask. So motilin levels are maximum just prior to gastric onset phase 3 MMCs, but not duodenal. So let's just break that down real quick. It's super simple. They're maximum just before phase 3 of the MMC in the stomach. But if you have an MMC that's not in the stomach and is in the duodenum, then it does not increase. So the duodenum has its own MMCs and the stomach has its own. The stomach has MMCs that it has by itself, right? And in these stomach MMCs in phase 3, which is the intense peristaltic activity, just before it, we're going to have maximum motilin levels. All right, super simple, just something you need to memorize and put in your head. The housekeeping function of the MMC. So during phase 3 antral MMC, which is in the stomach, obviously, because antrum is in the stomach, the pylorus relaxes markedly. The strong contraction leads to sweeping of roughage and any residual material from the stomach to the duodenum, which means anything that's left over is swept into the duodenum at stage 3 of the MMC, phase 3. It relaxes so much that even foreign bodies like small batteries and coins can enter the duodenum. So remember how I said the stomach doesn't let large particles enter? So what if you have something stuck? Like people swallow coins, batteries, all this stuff. And usually you see them later, it's in their feces, like they excrete it. So how did it get all the way to your rectum? And it's because during the phase 3 of the antral MMC, the pylorus opens so much that even foreign bodies can pass through. It also clears bacteria from the stomach. So how is MMC interrupted? MMC is during fasting. So how does it stop? It stops by feeding, because MMC during fasting, you stop fasting, you eat something, and then MMC is blocked. I mean, if you eat, right? Now we have gastric emptying, and this is the last concept in the entire lecture, and we're almost done. So gastric emptying is via antopyloroduodenal coordination. That's a very nice tongue twister. So anto for antrum, pyloro for pylorus, and duodenal for duodenum, because all of those are in a line. So this is the stomach. Just like imagine this is the stomach, antrum, this is the pylorus, and then we have the sphincter, and then we have the duodenum. So they're in line, right? So if these three things coordinate, and then only then can we have gastric emptying. So the antrum must contract to push the food towards the duodenum. The pylorus must be open, it must be relaxed, it must not be closed. Because if it's closed, the food's going to stay where it is. And the duodenum must relax to let the food enter. It should not contract. So if these three conditions are met, that's called antopyloroduodenal coordination. And only if we have antopyloroduodenal coordination, only then can we empty the stomach. During emptying, the fundic accommodation tone decreases and emptying tone increases. So this is the fundus of the stomach. It's superior to all of these. It also stores food, remember? So instead of storing the food now, its accommodation tone decreases. That means it doesn't want to accommodate the food anymore. And emptying tone increases. That means it wants to empty the food. And hence it will increase the vagal tone to contract so that it can move the food out of the way to the antrum. And then the antrum can do the antopyloroduodenal coordination and empty the food. The pyloric relaxation of the sphincter and the pylorus is via VIP and NO, and I've already told you these are relaxants always. Nitric oxide and basointestinal peptide, right? Okay, so now we'll review real quick. What happens? How does gastric emptying happen, and what slows it down? So if we have an increased tone in the ORAD region, the ORAD region is fundus plus proximal body of the stomach. So the fundus plus the proximal body of the stomach, which is like, let's say it's like this, no, actually this part, the line here, that's the ORAD. And if you have increased tone, obviously that's going to push the food towards the antrum, where it can undergo antopyloroduodenal coordination. Increased peristaltic contractions in the caudal region, okay? So obviously you want the peristaltic contractions to increase so that it can move out of the way. Decreased tone of pylorus. The pylorus must be open, and this happens via VIP and NO because those are relaxants. If the pylorus is relaxed, it can allow the food to pass through easily. And decreased contractions in duodenum, because you don't want there to be pressure in the duodenum, because then it's going to push the food backwards. You want it to be calm, You want it to be relaxed so that it can enter the food easily. Conditions slowing gastric emptying. Now, just everything that we had over here, flip it. If you flip it, it's going to be the opposite, and the opposite is slowing it down. Super simple. Enterogastric reflex. Okay, this is the last one. And then we're done. Alhamdulillah. I know it's super late. I'm sorry, guys. But I promise it's going to be worth it when you get an A in GIT. Enterogastric reflex is basically an intestinal reflex. So remember, we have the gastric emptying intestinal phase, which is the phase where the intestinal mechanoreceptors stop the gastric emptying because there's no more food left. So the intestinal phase starts when the stomach empties the chyme into the duodenum, and a reflex sustains further gastric emptying via the mechanoreceptors. Now the lipids, so we have the mechanoreceptors. We also have three other things. We have the CCK, which we studied earlier. We have the secretin, and we have lipids. We already studied CCK and secretin. Okay, we had these earlier. They're released by the I and the S cells, and they activate the afferent with an afferent vagal, which is the bad one. It's the bad guy, and this increases somatostatin and inhibits gastric secretion, right? So the lipids inhibit gastric emptying. And why do they inhibit gastric emptying? Well, it's because the lipids are hydrolyzed into fatty acids continuously, whereas the other things such as the acid are neutralized by the pancreatic bicarbonate. The osmotic stress is countered via the movement of water across epithelium, and the irritants are also countered. But lipids are broken down into fatty acids, right? And fatty acids cannot be broken down in the stomach. They can only be broken down in the intestine. So these fatty acids, they irritate or inhibit the gastric emptying because there's no, they cannot be stopped. We don't have a means to stop them until we reach the intestine. So the more lipids you eat, the more the gastric emptying is stopped, which is why when you eat something that's very fatty, you're going to see the gastric emptying is going to be stopped. So it says something very fatty is going to stop gastric emptying because of this reason, because the fatty acids are going to annoy, quote unquote, the stomach, and it's not going to be emptied quickly. The other two things are CCK and secretin. CCK is released by I cells, secretin is released by S cells. We already did this. And they lead to an release of and irritants. Obviously, irritants can be anything. They release serotonin, and obviously this is controlled via the enteric nervous system. So CCK, secretin, and serotonin produce the vagal response. Look, guys, I know you're going to hate me so much, and I'm sorry, this is not my fault. But in the first part, CCK, secretin, and CCK and secretin activated afferent vagal, right? This is for the gastric. This is for gastric secretions, right? But when we're talking about gastric emptying, in that case, CCK, secretin, and serotonin, the same ones, they activate vagal. Okay. Please don't make a mistake in this because I feel like this is something that's confusing and it's easy to make a mistake. But literally just 1 second, just listen to me. CCK and secretin in the gastric secretions activates afferent vagal, which activates somatostatin and leads to an inhibition of gastric secretion. This is in gastric secretions. When we're talking about gastric emptying, on the other hand, emptying, which is like a mechanical movement to empty stuff, the CCK, secretin, and serotonin activate the vagal reflex. Okay? And the vagal response produces these changes, which is a decreased fundic. Vagal is basically going to want to cause the food to not go. It's going to want the food to stay there. Okay, so what do you think? We talked about the antopyloroduodenal thing, right? We need the antrum to be contracting, the pylorus to be relaxing, and the duodenum to be relaxing. So now we don't want the food to go because the vagal is saying no, don't let the food go. So in this case, we have the opposite. So decreased fundic tone. Okay, so we're going to have NO and VIP, which is going to contract the fundus. We're going to have we're going to have NO and VIP, which relaxes the fundus, which means there's not going to be contraction and pushing it towards the antrum. So the food is going to stay in the fundus. Decreased antral peristalsis, so no contraction of the antrum. Increased pyloric constriction, which means the pylorus is going to close itself, and increased duodenal peristalsis, which means the pressure inside the duodenum is going to increase, which means that it's not going to allow food to come in. So basically, the opposite of whatever enables gastric emptying. Whatever enables gastric emptying is going to inhibit it here, and it's through the vagovagal response. The vagovagal response does not want the food to go into your stomach. I'm sorry, I know there's so much vagovagal vagus when you haven't done the vagus nerve properly, but that's just how it is, unfortunately. So in addition to this, obviously the lipid disrupts the antropyloduodenal coordination further, which inhibits gastric emptying even more. As I said, the lipid is the most important inhibitor of gastric emptying because it irritates the stomach by the fatty acids and inhibits antropyloduodenal coordination. Okay, last slide, which is very super simple. It's something you've already studied, and we'll just go over it really quick. Lag time. So if you eat a liquid meal, which is this one, it's going to be digested quicker. If you eat a homogenized solid, which means something that's uniform, it's going to take some more time to digest, but still pretty quick. Low-fat solid is going to take more time to digest, and then high-fat solid is going to take so long. Look at this lag. Oh my God, this much. Okay, so basically, if you eat a meal that's more fat, it's going to take more time to empty the stomach because gastric emptying is inhibited via the gastric emptying is inhibited via the lipids, obviously, and lipids are fats. So if you have more fats, you have more lipids, which inhibits the gastric emptying and causes a lag. But if you have a liquid meal that has no fat, it's going to have no lag, right? And that's all there is. And please scan this. If you scan this, I promise you're going to get an A. And there's only one question in this, so you can just fill it out. It's going to take like 10 seconds. I already know I took so much of your time. Like, we've been here for almost 2 hours. So I apologize for taking so long, but I promise that I've covered everything, and inshallah, this should be good enough. Anyone has any questions? I'm here, and we're done now. So we can stop recording.